Hey everyone, welcome to this explainer. I'm absolutely thrilled to be your guide today through the ultimate exam blueprint on excretion and osmoregulation. We're going to take a highly visual, scannable journey right through the biology of waste removal, and we're going to do it with a lot of energy. If you're prepping for your advanced high school, FSC, or Cambridge A-level exams, this content is perfectly engineered for you. Let's get right into it.
Okay, let's dive into this. Here's our syllabus blueprint for today. We're looking at 6 key areas: the core definition of excretion, the kidney and the nephron, the exact sequence of forming urine, homeostasis and temperature control, plant and invertebrate adaptations, and finally, a rapid-fire 10-second exam summary. Let's go.
All right, kicking things off with Section 1, the core, excretion defined.
Think of every single living cell in your body as a tiny microscopic factory. And just like any factory, all those chemical reactions, they produce waste. If a cell doesn't get rid of this toxic trash, it's just not going to survive. So excretion is literally the elimination of these metabolic waste products from the body. We're talking carbon dioxide, excess water, salts, urea, and uric acid. The absolutely crucial word to remember here is metabolic. Excretion deals specifically with toxic byproducts created inside the cells during chemical reactions, like cellular respiration or breaking down amino acids.
Now, stop right here, because we really need to address a massive exam trap. Excretion is the elimination of cellular metabolic waste. Egestion, or defecation, that's just getting rid of undigested food from your digestive tract. Look, feces were never actually inside your cells. They just passed through the tube of your gut. Mixing up these two biological processes is a classic mistake. Excreting cellular waste is completely, fundamentally different from defecating undigested food. Lock that distinction in right now.
Now, what's really interesting about this slide is how an organism's environment strictly dictates the kind of nitrogenous waste it produces. Let's look closely at ammonia. It's highly toxic, right? It needs a massive amount of water to safely dilute it, so naturally it's excreted by aquatic organisms like bony fish. Then we have urea. It's moderately toxic, needs a moderate amount of water, making it perfect for terrestrial animals like us mammals. But uric acid? This is brilliant. It's the least toxic and can be excreted as a nearly dry paste. It's literally the ultimate adaptation for dry environments, used by birds, reptiles, and insects to heavily conserve their precious water.
Moving right along to Section 2, the kidney and the nephron.
Your kidneys are your body's ultimate biological purifiers. They're absolutely critical for keeping your blood clean and your blood pressure stable. Notice the renal artery there? It's bringing waste-heavy blood directly from the aorta right into these fist-sized, bean-shaped organs. And your kidneys, they never take a break. They're constantly taking that high-pressured blood, regulating the exact water content, and pulling out toxic metabolic wastes, like urea, to send straight down to the bladder.
Okay, heads up, here's another absolute exam trap to watch out for. The ureter versus the urethra. The ureter, spelled with an E, connects the kidney down to the bladder. The urethra, with an A, connects the bladder to the outside of the body. I know they sound super similar, but functionally, they're completely different tubes. I can guarantee examiners are going to try to catch you on this, so make sure you never mix them up on test day. All right, let's zoom in on the internal anatomy. You can clearly see the distinct outer layer, the cortex, and the inner layer, the medulla. But the real magic? That happens right at the boundary between the two, in a microscopic structure called the nephron. You can kind of think of the nephron as a microscopic coffee filter for your blood. You've got about 1 million of these functional units packed into each kidney, doing all the actual heavy lifting of excretion and osmoregulation. Which brings us to Section 3, sequence, forming the urine.
So we're going to take the complex mechanism happening inside that tiny nephron and break it down into scannable, logical steps. There are 3 chronological milestones you absolutely must know. Step 1 is ultrafiltration. High blood pressure literally forces small molecules out of the capillary knot, that's the glomerulus, and into the Bowman's capsule to form a filtrate. Step 2 is selective reabsorption. Now, the body isn't foolish. It actively returns useful things like glucose, water, and essential salts back into the blood so they aren't lost. Finally, step 3, tubular secretion. Here, the body uses active transport to forcefully move any stubborn remaining wastes, like uric acid and hydrogen ions, from the blood directly into the tubule to guarantee they leave the body.
And this brilliantly illustrates exactly how effective that sequence is. Just look at the concentrations in the blood versus the urine. Urea jumps from a tiny 0.03% way up to 2%. Now that is highly concentrated waste.
But look really closely at proteins and glucose. They're heavily present in the blood, but completely absent, 0% in the urine. Why is that? Well, because proteins are way too large to ever be filtered out in step 1, and glucose is 100% selectively reabsorbed in step 2. If you see proteins or glucose in urine on an exam question, you immediately know there's a major biological failure happening.
Let's shift gears to Section 4, homeostasis and temperature control.
You know, excretion isn't just some isolated event. It's a huge part of homeostasis, the maintenance of a constant internal environment. Notice those coiled sweat glands located deep in the dermis layer? The skin actually acts as a secondary excretory organ. Through those sweat pores, we excrete excess water, urea, and salts. But here's the cool part. While it's getting rid of waste, this exact same mechanism is simultaneously working to tightly regulate our core body temperature.
Let's contrast how the body involuntarily responds to extreme environments. In cold weather, you experience vasoconstriction. Your blood vessels literally narrow to actively pull blood away from the skin surface to save heat. You shiver, and your hair erector muscles contract to trap an insulating layer of air. But in hot weather, it's the complete opposite. Vasodilation widens the vessels to radiate heat away from the skin, and sweating increases dramatically so that the water can take that heat away through evaporative cooling.
All right, Section 5, plants and invertebrate adaptations.
It is incredibly fascinating to see how completely different life forms, ones that don't have human kidneys or skin, handle the exact same problem of metabolic waste. Plants have totally different strategies from us. First off, they have a lower overall metabolic rate, so they just produce less waste to begin with. Plus, they synthesize exactly the organic food they need, meaning there's no excess protein to break down into toxic nitrogenous waste. Instead, they store what little waste they do have in large vacuoles. But honestly, the most brilliant adaptation? Storing waste in their leaves. When autumn comes around, those yellowing, falling leaves act exactly like the plant's garbage bags, brilliantly disposing of accumulated toxic heavy metals as they drop to the ground.
Now, take a look at insects. They utilize a really unique structure called the Malpighian tubules, located right between the midgut and the hindgut. Because insects often live in dry terrestrial environments, they really can't afford to lose any water. So these tubules creatively excrete nitrogenous waste as solid uric acid crystals right along with their feces. It's just a highly efficient way to clear toxins while hoarding every last drop of precious moisture.
But wait, what if your problem is actually too much water? Take the single-celled amoeba living in a hypotonic, freshwater environment. Water is constantly rushing into it by osmosis. To survive this microscopic flooding, the amoeba uses a structure called a contractile vacuole. You can think of it like an active bailing pump on a sinking boat. It constantly collects that excess water and forcefully pumps it back out through the cell membrane so the poor organism doesn't literally burst.
And finally, Section 6, the 10-second exam summary.
Let's ramp up the energy, pull all these threads together, and lock in these high-yield facts. So the crucial point is, if you remember absolutely nothing else from this explainer, remember these 4 bullets. 1. Excretion is purely metabolic waste. Defecation is just undigested food. 2. Know your habitat rules. Ammonia is aquatic, urea is terrestrial, uric acid is for dry environments. 3. The nephron functions in a strict, non-negotiable order. Ultrafiltration, then selective reabsorption, then tubular secretion. And 4. The ureter connects the kidney to the bladder, while the urethra connects the bladder to the outside. Master these, and you are perfectly prepped for your exams. I want to leave you with a final, provocative thought today. If our biological kidneys are perfectly engineered to filter microscopic waste, actively reabsorb nutrients, and strictly regulate water, how might the future of bioengineering create fully artificial, implantable kidneys? Could we completely eliminate the need for dialysis machines in our lifetime? It's a massive biological challenge just waiting for the next generation of scientists to solve. Keep studying, keep questioning, and I will see you in the next explainer.